Okay, so let's get started. Hello, everyone. Welcome to our today's Immune Zoom seminar. So it's really my pleasure to welcome our today's speaker, Dr. Wei from Indiana University School of Medicine. So Wei currently is assistant professor in Department of Microbiology and Immunology. So Wei, he actually completed his undergraduate study in China, in China Pharmaceutical University in Nanjing, and then he moved to United States to pursue his PhD training. He actually received his PhD in biochemistry from sunny Buffalo for his work that identified the pathway connecting transcription factor ETS1 to B cell mediated autoimmunity. Um, then he moved on and completed his first postdoc training at the University of Pittsburgh, focused on defining the important signaling pathway regulating germinal center B cell selection. And then his second postdoc fellowship at Stanford mainly focused on B cell responses to vaccination and developing better vaccine against influenza. Now, from 2021, Wei actually started his independent research program and started his lab at Indiana University as assistant professor to continue his work on B cell biology. His group is currently trying to connect the mechanisms regulating germinal center reaction to vaccine design. And then the main goal of his research program is to develop vaccines which can induce broad and durable B cell memory against a deadly infectious disease. On the other hand, uncontrolled B cell response may also lead to autoimmune disease such as lupus and then rheumatoid arthritis. Um, his lab also is interested in understanding the pathogenesis of these autoimmune disease to help to develop new therapeutic approaches. So I think Wei actually are going to talk about his B cell selection work for us today, and then we're really excited to look forward to your seminar. Please go ahead.
Yeah. Thank you very much, Chuan and Paul. Minimize this. Uh, so I started this work when I was a postdoc at Mark Stanchik's lab. So I started this about 10 years ago. I'm very lucky when I started my own lab, I was able to continue some of the studies with Mark as a collaborator. Actually, even though I did most of the experiments, but I almost never had a chance to present this work to a broader audience. I really appreciate this opportunity to be able to present this work. Thank you, Paul and Chuan, for having me. The title of the talk is Signaling Guided Germinal Center B cell Selection and Fit Decisions. So first, let's compare germinal center response with actual follicular response. So when B cell meets the antigen, either when we get a vaccination or we get a pathogen like a virus, there are two pathways the B cell can mount an antibody response. The first pathway is pretty fast. So B cells depends on the signaling and the microenvironment. It might directly differentiate into effector cells, which are the short-lived plasma cells or memory B cells. So the short-lived plasma cells can make the first wave of antibody response to be able to control the infection. And usually the antibodies made by the actual follicular response is low affinity and mostly IgM, even though there are some IgG antibodies can be made. And at this early time, the B cells are trying to control the disease. So sometimes they will make autoantibodies. But most of the time, it's not a concern because the autoantibodies will go away very quickly because the short-lived plasma cells will die very, very quickly. So the second route of response is the germinal center. So with the T cell help, some B cells will come to the germinal center and start the germinal center response. The germinal center has dark zone and light zone. So B cells in the dark zone, they proliferate, and then they translocate to the light zone to interact with other immune cells and antigens presented by follicular dendritic cells. And eventually, they will differentiate into memory B cells and long-lived plasma cells. So compared to actual follicular response, the germinal center response usually generate antibody of very high affinity, and mostly the antibodies are class switched to IgG or IgA because it's called long-lived plasma cells, and they can migrate into the bone marrow and secrete antibody for extended period of time. Sometimes it can be decades. So it can provide very durable protection, and it's more stringent, which means in germinal center they usually don't generate autoantibodies because the autoreactive B cells are selected against in germinal center response. So the germinal center is very unique. The first point is the affinity-based selection. So germinal center B cells express a protein called AID, and AID can introduce mutations to the B cell receptor, which are essentially antibodies on the B cell surface, and they use it as a receptor to sense the antigen. So when the mutations happens to the B cell receptor, there are uh, multiple outcomes can happen. The first is it might generate a higher affinity B cell receptor, or because the mutation is random, it can also damage BCR or generate a lower affinity B cell receptor. And occasionally, it can generate a B cell receptor that can bind to autoantigen and potentially become autoreactive. So the cells will compete with each other, and eventually, the lower affinity or the autoimmune B cells they will die unless autoimmune B cells can mutate away very quickly from autoimmunity. Otherwise, it will die. So for the higher affinity B cells, it will start express a protein c-Myc, and this will mediate their proliferation. So in germinal center, the B cell will go through this selection with many rounds, and eventually the affinity will become really high. So the amazing thing is for germinal center, depends on immunization strategies. It can last the time of a germinal center can last for either weeks or sometimes to months or even over a year. Like if you shown by polypolandrin, if you use a very strong adjuvant or Shincroti, Dario or Irvin, if you reformulate the antigen for sustained release, sometimes the germinal center can last for 6 months or even a year. So during this time, the affinity maturation just keep happening, and eventually it can reach over 1,000 folds. So this is facilitated by the clonal evolution in the germinal center. Another, which I think is quite amazing thing about germinal center is they continue produce effector cells without showing any exhaustion. So germinal center can produce long-lived plasma cells. So my lab mate Florin a few years ago published a paper in this publication. He used BRDO to label the cells, so he can track the plasma cell or uh, memory B cell differentiation in real time. So he used a NPCGG immunization strategy, which induced a germinal center peak around like day 12 or day 14. So this is around the peak time of germinal center. But what he found is even though the germinal center starts to decline after 3 or 4 weeks, but the germinal center continue to pump out long-lived plasma cells. This doesn't show much decline until the very end of the germinal center response. For memory B cells, it's similar, even though different subsets of memory B cells are generated at different time point. For example, this CD80 PD-L2 double negative memory B cells are generated very early on and followed by the single positive. But the CD80 PD-L2 double positive memory B cells are actually they are continuous generated throughout the germinal center reaction, which is in this immunization strategy, it's about 1 or 2 months the germinal center can last. So this suggests even though the germinal center they can last for extended period of time, they don't they unlike for example T cells in chronic viral infection or in a cancer, they enter the cancer or in the chronic viral infection, the CD8 T cells quickly get exhausted. For germinal center, somehow it can manage and they can still carry out their effector function, and they can continuously doing affinity maturation and pump out memory compartments, long-lived plasma cells and memory B cells. Even if it can last for a year and it can still doing this. So it's quite amazing. And the question is, how does germinal center regulate this, right? How this is possible? So this work, I was inspired by some prior works showing that if you compare germinal center B cells, this is central blast and central sites are dark zone germinal center cells and light zone germinal center cells. If you compare them with naive B cells or memory B cells, you can see, the naive and memory B cells, they are quite similar in terms of gene expression profile involving the signaling. But the germinal center B cells are very, very different from either naive or memory B cells, suggesting even though there are different signals can happen to naive germinal center or memory B cells, maybe the signaling pathway or the regulation of this different signaling is different between germinal center B cells and naive or memory B cells. So to study this, I use a strategy that's pretty straightforward. Just directly compare primary germinal center B cells with naive B cells. There are two approaches I used in most of the following studies. Essentially, I immunize the mice, which are B18 or mic mice. So those are transgenic or knocking mice, so they naturally carry some B cells that has some affinity against nitrophenol. So I can use this model antigen NPCGG adjuvanted by alum for immunization, and it will induce robust germinal center response. So I either just stimulate splenocytes or purified total B cells, and then use flow cytometry, so get on germinal center B cells or non-germinal center B cells, and directly compare these two populations for their signaling events. Or in order to do more experiments like RNA sequencing, I can purify naive B cells from a naive mice and purify the germinal center B cells from immunized mice. And I developed a beads-based purification that's very gentle and doesn't affect the cell signaling, and I can reach extremely high purity for germinal center B cells. Then I stimulate the cells with defined signals and study how they respond to the signals using different experiments. So based on this, I did a few experiments. I quickly found out, yes, indeed, the germinal center B cell signaling different pathways are all reprogrammed. For example, the B cells, when they encounter antigen can bind to the B cell receptor and generate BCR signal. For naive B cells, if you uh, stimulate B cells with antigen, and you can generate a robust phospho-Syk. This is proximal B cell receptor signaling, and the downstream is phospho-S6 and IL-4, which IL-4 is a downstream of NF-kappa B pathway. And you can see the naive B cells is robust signaling. I mean, germinal center B cells, it's very little phospho-Syk and essentially no downstream signaling generated. For CD40, it's quite similar. If you stimulate naive B cells with anti-CD40 antibody, it can generate a very good phospho-AKT, phospho-S6, or phospho-ERK signal. But for germinal center, it's all attenuated. These signals are all attenuated. For IL-21, it's a little different. So IL-21 can induce JAK-STAT signaling and eventually phosphorylate the STAT3 at the tyrosine 705. And for naive B cells, and you can see when you stimulate with IL-21, and you can see a robust signal of phospho-STAT3, 15 minutes to 30 minutes, and it can last for 2 hours or 4 hours. It can really persist. However, for germinal center, it's interesting. You can induce a very robust phospho-STAT3 at 15 and 30 minutes, but it's very transient because at 2 hours it goes down, 4 hours it's almost back to normal, to the baseline. So it's the kinetic is very different from naive B cells. So the signaling pathways in germinal center B cells are indeed very different from what we learned for the naive B cells, which is very similar to naive B cells and the memory B cells. They are similar. So you that leads to some questions like if the germinal center like K signals in germinal center is attenuated, how do they carry out those functions, affinity maturation and the effector differentiation? So there are two discoveries that's very intriguing at that time. First is uh, Dalla Faber lab and the Rojaski's labs. They found that FOXO1 controls the germinal center zonal transition, which means the FOXO1 has to be turned off in order for germinal center B cells the translocate from the dark zone to the light zone, which is essential for them to interact with other immune cells. Uh, Another finding is that CMYC is very critically regulate positive selection of germinal center B cells. Still, Dalla Favra and Rojaski's group suggest that CMYC is the K selection marker for germinal center B cells, and Hodgkin group showed that CMYC is like a timer. If you have more CMYC, you can divide for longer period of time, which means CMYC is really essential for controlling B cell proliferation. This, so the model, the picture is now look like this. So the B cells in the dark zone needs to inactivate FOXO, which usually carried out by a phosphorylation, and then in the light zone, somehow it can turn on the CMYC expression in order to get recharged to proliferate. So the first question is what signals are regulating the inactivation of FOXO or the activation of CMYC? And the third question, of course, is what signals regulate the balance between germinal center, return remain in the germinal center, or they finally decide to exit germinal center and differentiate into memory B cells or long-lived plasma cells. So to answer these questions, we first focused on signal one, which is the antigen binding to the B cell receptor. So for germinal center B cells, even though the phospho-Syk is really attenuated, but you can see there is a solid, there is a tiny little shift. And so I did a track the kinetics of phospho-Syk and its downstream signals. What I found is even though this phospho-Syk is very small and transient, but eventually it can lead to a almost normal phospho-AKT signal at just 1 minute, because at 3 minutes, for naive B cells, the phospho-AKT just stays on, but for germinal center, it's only just 1 minute of phospho-AKT. And apparently, this very transient phospho-AKT or phospho-Syk cannot lead to a very good phospho-S6 or phospho-ERK signal. But however, surprisingly, this transient AKT can really phosphorylate FOXO1, and you can see germinal center B cells naturally express a lot higher levels of FOXO1, but still AKT is able to very efficiently phosphorylate FOXO1 because FOXO1 is a direct target of AKT signal. And this leads to a very efficient uh, exclusion of FOXO1 from nucleus. As you know, FOXO1 is a transcriptional repressor. So once in the nucleus, it's active. And all it takes is 5 minutes of anti-IgM, which stimulate the B cell receptor, for FOXO1 to be excluded from nucleus and goes to cytoplasma. So this suggests for naive B cells, the B cell signal is a multifunctional. It can lead to activation of different pathways. But for germinal center, it can only go through this route of transient phospho-Syk active AKT and inactivate FOXO1. And this is essentially a 5 minutes event. So FOXO1, as you know, if it directs germinal center zonal translocation, right? So if a BCR signaling can inactivate FOXO1, whether it can help germinal center B cells translocate from dark zone to the light zone. To study this, we essentially use a inducible Curie system. In vivo, we can give tamoxifen to the mice and knock out the Syk, which is the proximal BCR signaling molecule, and then you can eliminate or reduce the B cell receptor signaling. What happens is compared to control mice, only 2 days of Syk deletion in established germinal center is able to reduce the light zone. As this is the light zone area, and you can see this is a robust reduction. And 2 or 3 or 4 days tamoxifen treatment deletion of Syk can reduce germinal center number, probably because it cannot translocate to the light zone to interact with T cells. So the first problem is kind of resolved. So it's the B cell receptors signaling that can help the B cells from dark zone to the light zone in order to interact with other cells. So what about the second question? What signals are actually induce c-Myc in germinal center B cells, which is very important for their positive selection? So we go to the signal two, which is a CD40 ligand from T cells binding to CD40 on B cells. Again, the signaling is attenuated, seemingly attenuated. And uh, unlike the B cell receptor signal, CD40 stimulation in naive B cells can really inactivate FOXO1 very quickly. But for germinal center, there is no sign of translocation of FOXO with CD40, so it cannot lead to CD40 FOXO inactivation. However, when we look at NF-kappa B signal, and this is a germinal center, and you can see anti-CD40 stimulation for 30 minutes can effectively activate NF-kappa B. So when it's activated, of course, it's different from FOXO, so it's translocate from cytoplasma into the nucleus, meaning the NF-kappa B is activated. So similarly, naive B cells, CD40 can activate multiple signaling pathways, but for germinal center, only NF-κB is activated, not AKT, FOXO, or ERK signaling is activated by CD40 in germinal center cells. So this has become very interesting because for naive cells, either B cell receptor or CD40 signal can lead to multiple pathway activation, which induce NF-kappa B pathway and inactivate FOXO1. So c-Myc is actually activated by NF-κB and inhibited by FOXO1. So if you want a robust c-Myc expression and you want a active NF-κB signaling but inactive FOXO1 in order to release the inhibition. For naive B cells, either a B cell receptor or CD40 can do both, right? So if you stimulate naive B cells and c-Myc is robustly induced, you just need one of the two signals. It can induce c-Myc expression. But for germinal center, because BCR signaling is restricted to this pathway for FOXO1 inhibition, and the CD40 can only activate NF-κB, that leads to a hypothesis that this enforce a collaboration of both B cell receptor and CD40 in order to induce c-Myc expression. And that's exactly the case because if you, for germinal center, if you stimulate them with one signal, it can barely induce c-Myc. But if you combine both, there is a synergy and you can robustly induce c-Myc expression. And it also leads to a synergistic induction of phospho-6, which is another positive selection marker for germinal center B cells. So we validate this in vivo. The first experiment is how about we just give a strong B cell receptor signal. So we need a special mouse line, which is IgMI mouse. And they don't have circulating antibodies, so you can inject a anti-IgM and they can reach germinal center B cells without binding to the circulating antibody. And this 4-hour stimulation, and you can robustly induce c-Myc expression in germinal center B cells in a small population of germinal center B cells. This is a lot less compared to naive B cells, presumably because this population probably get a CD40 signal very recently, right? But a strong B cell signal definitely can induce c-Myc expression in germinal center B cells. So another way to validate this is, so we asked a question. So without a valid B cell receptor signaling, whether a very strong CD40 can overcome the lack of B cell signaling to induce c-Myc in germinal center B cells. So to do that, again, we use this tamoxifen inducible Cre system. So we can delete the B cell receptor signaling in ongoing germinal center response by delete the Syk molecule. And after that, we IV inject anti-CD40 antibody which can introduce a very strong CD40 signal to germinal center B cells. So 4 hours later, we compare naive B cells and germinal center B cells. So for naive B cells, it seems as we expected, they don't really care whether they have a B cell receptor signal or not. A CD40 signal is very strong, is strong enough to induce c-Myc in almost all the naive B cells. But for germinal center, it's very different. So with for the wild type, with the Syk can transduce B cell receptor signal, the CD40 can induce c-Myc in a subset of germinal center B cells. Presumably, those germinal center B cells are getting a BCR stimulation or antigen stimulation around the same time. But without the B cell receptor signal, even this anti-CD40 very strong CD40 stimulation cannot induce any c-Myc in germinal center B cells. So this two in vivo experiment really validate a germinal center will need a collaboration of both B cell receptor and CD40 signal for their positive selection. So the picture now is more intact. The dark zone germinal center B cells will need to bind to the antigen. Presumably, these antigen are soluble antigen, not deposited in the follicular dendritic cells. And this BCR signal in active FOXO1 so the B cells can translocate to the light zone in order to interact with the follicular dendritic cells and T cells. So follicular dendritic cells give antigen-derived B cell signal to keep the germinal center B cells in the light zone, and then to receive T cell signal. And T cell gave the B cell CD40, and both the B cell receptor and CD40 signal are required for robust induction of c-Myc and phospho-6 to recharge the germinal center B cells for proliferation. And when they proliferate, they slowly use up their battery, and then they need to go through this again. And after rounds of selection, and you can imagine the affinity will quickly increase. So the final question, the final puzzle is what signals direct germinal center exit and effector differentiation into memory B cells and long-lived plasma cells? So, of course, we go to signal 3, which is L21, and binding to derived from T cells and binding to L21 receptor. So, uh, as I showed you before, even though germinal center B cells can have a valid L21 receptor signal in terms of phosphorylation of STAT3, but the STAT3 doesn't last, right? It only lasts for about 30 minutes. For 4 hours, and you can see L21 alone, the phospho-STAT3 is still going in naive B cells, but barely detected in germinal center B cells. However, if you add a CD40, it seems like the phospho-STAT3 can sustain. This make me think maybe L21, the CD40, they all have some sort of synergy for germinal center B cells. So to do this, we don't want to forget about BCR with CD40. And so we compare what if the germinal center get a BCR signal with CD40 or get a CD40 plus L21. So these are labeled here. So again, naive B cells, they can robustly induce c-Myc with almost just one signal. And for germinal center, you need these two signals, and it seems like both BCR plus CD40 or L21 plus CD40 can give you c-Myc expression as well as phospho-6. So in terms of positive selection markers, there's almost no difference between the combination of these signals. However, when we did a RNA sequencing with stimulated germinal center B cells by different, we did all the different stimulation, but if you pay attention to the no stimulation and L21 CD40 and LGM anti-IgM, which is BCR plus CD40 signal, even though they both induce c-Myc and phospho-6, but by this PSA plot, you can easily tell they are going different route, right? The L21 CD40 2 hours and 4 hours they are going up, and apparently this BCR plus CD40 going down. This really suggests even though they both mediate positive selection, but maybe the outcome is different for these different combinations of signals. And another way to look at this is you look at this plot. You put the BCR CD40 on x-axis and L21 CD40 on the y-axis, and anything aligned in this triangle line are essentially genes are regulated similarly between these two different combinations. However, there are some signals, for example, toxo2 and toxo, it can be uh, increased with L21 plus CD40 signal, but it doesn't change with IgM plus CD40 signal. There are a bunch of other genes, this transferrin factor, this is non-transferrin factor. This really suggests this L21, the BCR combination with CD40, they're doing very different things to germinal center B cells. So what is the difference? So we want to look first. We want to look at the plasma cell differentiation. So the germinal center B cells will need a CD40 signal in order to survive. Without CD40, they die within hours. But with CD40, they can survive for 48 hours. And for 48 hours allows them enough time for them to differentiate. So, So we tried different combination, CD40L21, CD40 NB FICO, which stimulate the B cell receptor, and we also have triple stimulation. First, we look at the B cell 6 with BLIMP1 because everybody knows BLIMP1 is a plasma cell marker. And we think if you just look at this plot, right, you would probably think the B cell plus CD40 induce a lot more, or this triple induce a lot more BLIMP1, so they must be differentiating into plasma cells. However, this is not the truth. This is another example how germinal center B cells break the rule that what we normally think of B cells. So if you get on this BLIMP1 positive B cell 6 low population and look at some other plasma cell markers like CD138 and IL4, what you can say is even though this L21 plus CD40 can generate a little bit less BLIMP1 positive cells, but these cells are actually differentiating into plasma cells with 20.5% in this case. And strangely, the B cell plus CD40 induce more BLIMP positive cells, but those cells are not differentiating plasma cells. They can't further abrogate, if you draw a line, so I draw a line here, so they can't further upregulate IF4, which is essential for plasma cell differentiation. That's probably why they really stop here. They can't really induce CD138 and fully differentiate into plasma cells. And this is the quantitation. You can clearly say L21 CD40 is the only stimulation can robustly induce the plasma cell precursor, because essentially those are still germinal center. They still have some B cell 6 expression. So they are robustly induced plasma cell precursor differentiation in germinal center B cells. And there seems to be a big difference between IgM and IgG. So we further look at compared IgM positive and IgM negative. In APCGG immunization, most of these cells express IgG1 on their surface. So if you look at this, BLIMP1, there's almost no difference. But if you get on this and look at their differentiation, you can see the IgG germinal center B cells are differentiating into plasma cells, but not IgM germinal center B cells. This really suggests there is a big difference between IgM and IgG. That also explains why we get plenty of IgG plasma cells in our bone marrow, and we eventually lose IgM antibody for the long-term humoral immunity. So as I shown you before, for germinal center B cells, they try to control their signal for multiple pathways. For me, I think this is a strategy for germinal center B cells to be not overwhelmed by too much signal that might induce exhaustion, right? For germinal center, for L21, it's similar. So the phosphostat rate comes down very quickly, and then they turn it off also very quickly. So what is the mechanism? So another question is whether we can enhance the plasma cell differentiation by manipulate this pathway if we can make L21 signal persist a little bit longer, can we enhance plasma cell differentiation? So we did a lot of work. For the sake of time, I only have a couple of slides showing this, but this paper is in press, come out very soon. Make a long story short, we found out is the P10, which is highly expressed in germinal center B cells, that can inhibit IL-21 signal and make this very transient. Because this is a flow plot, and you can see for naive B cells, with or without P10, if you stimulate with L21, the signal lasts for 30 minutes, 120 minutes, or 2 hours, similar. For germinal center B cells, for wild type, which is P10 intact, the 30 minutes, there is plenty of phosphostatin 3, but after 2 hours, it comes back, right? But if you delete P10 in established germinal center B cells, and the signal stays on, and this is the quantitation. So it seems like P10 is very important in kind of making the phospho-STAT3 very transient following IL-21 receptor signaling. So if we, so can we boost plasma cell differentiation to inhibit by inhibiting P10? So to test this, there are several uh, 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 difficulties to test this in a very rigorous way. The first is the plasma cells can also come from actual follicular response. So a lot of times it's hard to track the plasma cells that's derived from germinal center. Another problem is if you delete P10 too early, it can affect actual follicular response, which can generate a kind of different feedback in germinal center. So you will never know whether the effect is a germinal center B cell intrinsic. So to overcome these problems, so I have to develop this germinal center transfer or camera model system. So what I did was I purified germinal center B cells from a day 14 immunized mice. So those are peak of germinal center time point. I purify the germinal center B cells from CD45.1 or CD45.2 mice. And then I retrovirally transduced these cells with either CD8 shRNA, this is as a control, the B cell don't express CD8, or a P10 shRNA to knock down P10. And this P10 shRNA works really well, can knock down P10 really well. And then I transfer, I can transfer equal number of the transduced cells into a CD45.1 and point 2 mice. And 8 days later, I use FACS to analyze the mice germinal center response. And in this way, because I transferred already established germinal center B cells for any plasma cells marked by the congeneric markers will be from the germinal center cells. So what happens to germinal center? There is some advantage when you knock down P10 in germinal center cells, but not that much. But when you get on germinal center cells and then look at their plasma cell precursor differentiation by tracking IF4 and BLIMP1, and you can see this BLIMP1 knockdown can really boost plasma cell precursor differentiation, and this leads to a almost 10 times higher plasma cell differentiation as compared to the controls. Because these germinal center cells are in exactly the same microenvironment, so the only thing can affect their differentiation would be P10, and this is the plasma cell normalized to the germinal center B cells, and you can see there's definitely advantage of germinal center differentiation of plasma cells. So this really validates IL-21 signal is very important for plasma cell differentiation controlled by P10. So how about memory B cells? So again, I stimulated purified germinal center B cells with all different combination of signals, and what we found is the both CD40 IL-21 or CD40 or the triple stimulation can promote memory B cell differentiation as marked by higher expression of CD30 and CCR6. Again, there seems to be a difference between IgM and IgG memory B cell differentiation, and it's also depends. This is more complicated because it also depends on the combination of signals. So for IL-21 plus CD40 stimulation, it seems it promotes more IgM memory B cell differentiation as compared to IgG memory B cell differentiation. However, if you add the BCR stimulation into this combination, and then you get a lot more IgG1 memory B cell differentiation from germinal center B cells. So this is the quantitation. This suggests even though IL-21 plus CD40 can promote the plasma cell, IgG plasma cell differentiation, but you might need some BCR signal in order to generate more IgG memory B cells. So to summarize, the conclusion, the take-home message would be the relative strength of various signals may instruct the fit decision of germinal center B cells. And if you get a B cell receptor and CD40 for this, I want to be clear that the germinal center B cells in germinal center are likely getting all three different signals for any given time. It's more important is the relevant strengths because at different time points, what I'm going to talk about in a little bit is it's the relevance of all these different signals might direct them into different fate decision. So if it's a predominant B cell receptor plus CD40 signal, it can induce positive selection, but these B cells, they cannot effector differentiation and they remain as germinal center B cells. So for expansion of germinal center. For L21 receptor plus CD40 signal, if these two signal dominate, it's still positive select germinal center B cells, but the outcome would be it will promote the effector differentiation into plasma cells and memory B cells. And plasma cells is highly dominated by IgG because IgG germinal center B cells is more easily to induce IL-4 and CD138 upon this combination of signal. If you have additional B cell receptor signal, it can inhibit the IL-4 upregulation and it can promote BLIMP1 expression in germinal center B cell. As the outcome, it will inhibit plasma cell differentiation but promote the IgG memory B cell differentiation. So this is very interesting because if you think about germinal center from early germinal center, when you get plenty of antigen flowing around, there's more B cell receptor signal, but once the time goes on and the antibodies are generated and the antigens are slowly cleared, right? Then because antigen is less and less, then the B cell signal may become less and less dominant, and then the L21 signal may become dominant in this case. So from early mid to late germinal center, and because the signal combination has changed, they might have different outcomes. As early germinal center, the BCR plus CD40 will dominate germinal center expansion, while the early late they can output different memory B cells, especially for the late germinal center. It favors plasma cell over memory B cells, and this is again a Florence paper, and it's kind of show the similar thing. And late germinal center, you get the long plasma cell differentiation, while the memory B cells they peak in the middle and then they decline a little bit. And also inspires vaccine design about how can we balance these different signals to maximize the memory immunity development in order to protect us from pandemics. So that is this story, and this is my lab. I really want to thank my lab for supporting me during the last 4 years. And this is outdated picture. We probably need to update this. And I really want to thank my mentor, my friend, and my collaborator, Dr. Mark Stanchik. So I started all this work in Mark's lab about 10 years ago, and I'm so lucky. As of today, I can still collaborate with Mark. For all this project, there is more exciting science on the way. So I'm extremely lucky to have Mark as my mentor. And this is my founding sources. Thank you very much. And uh, if you have any questions, I'm happy to answer any questions. Thank you.